Hi, I see the numbers ticking up here, so we're going to go ahead and get started. Welcome to another Mass Medic webinar. My name is Nicole Owens. I'm the Director of Marketing Communications for the organization. If you're not familiar with us, we are a trade association for the medical device industry that works to bolster the New England ecosystem through education and connection, advocacy, and awareness. Today, we have a great webinar from our friends at Battelle. We're going to be talking about design differences between generic drug device combination products and reference listed drugs. Before we turn it over to our panelists for today, though, I do have a couple of housekeeping notes. So I just want to let everyone know that this webinar is being recorded, so you will get a copy of the recording after the webinar for everyone who registered. We're also going to be allotting time for Q&A. So as questions arise, please feel free to put them in the Q&A function at the bottom of your screen, and we will do our best to address all those questions as time allows. All right, without further ado, I'm going to turn it over to our panelists today from Battelle, David Herning. David is the Director of Human Factors and New Product Development for Battelle, and he's going to be sharing the presentation today. David.
Thank you so much, Nicole. And thank you all for joining me today. It's truly an honor to be able to share the research that we've conducted over the last 3 years related to assessing design differences between generic drug device combination products and reference listed drugs. As Nicole said, there we go. As Nicole has said, I'm the Director of Human-Centric Design and New Products Development here at Battelle. And just briefly, Battelle is a nonprofit. It's actually the world's largest nonprofit research and development organization. We were created by a charitable trust formed in 1925 with the single purpose to do the greatest good for humanity through science. It's exciting in that our profits go to really training the next generation of scientists and engineers, as we are a major supporter of the STEM education programs. Just a couple real quick disclaimers. The research was supported in part by a grant from the Food and Drug Administration of the U.S. Department of Health and Human Services. The views presented herein are those of the authors and do not necessarily reflect the official policies of the Department of Health and Human Services, nor does any mention of trade names, commercial practices, or organization imply endorsement by the United States government. The material that we're presenting today, or the material that really was the basis for the work that we're presenting today, is based upon both US regulations, international standards, and FDA guidance. You can see the references here on the screen. Excuse me. Generics play an important role in healthcare for a multitude of reasons, yet the process of bringing a generic to market is quite burdensome. Our goal was, when we started this process, and is to this day, to make the process of bringing life-supporting generic drugs available in a more timely and efficient manner for both manufacturers and regulatory agencies. A generic is expected to be therapeutically equivalent to an RLD. It's expected to produce the same clinical effect, have a similar safety profile, and be safe and effective. The one part, or the piece that is probably the strongest component, or the one thing to really remember, is it must be substitutable for an RLD without the intervention of a healthcare provider and or without any additional training prior to use. A generic is not expected to be an exact duplicate, and it often cannot be for various reasons. Just as a side note for today, I will be referring to generic drug device combination products as generics, and then their reference listed drugs or reference listed drugs as RLDs. The 2017 draft guidance states that a generic must produce the same clinical effect and have the same safety profile. In effect, it must be substitutable without any prior training or intervention of a healthcare provider. It's fascinating how the guidance strongly suggests that generic manufacturers should minimize user interface differences with the reference product. And it also implies that the only validation option is that of a comparative use human factors study. We're going to be looking at these a little bit more shortly. The current process carries a heavy burden for both generic manufacturers and regulatory authorities. Generic manufacturers appear to strive for replication, even though replication is not required, and in many cases is not really feasible. Stabilizing patient access can improve public health outcomes, reduce healthcare costs, and ensure that essential treatments are available to those who need them, regardless of their income or location. We knew when we started this process that we wanted to simplify the process to reduce the burden on both the manufacturer and regulatory. We wanted to minimize the dependence on the comparative use human factors study, and we wanted to improve the quality of regulatory filings so that these important generic products would be able to get to the market as quickly as possible. And then through that whole process, we got this thought, what if we could actually do better than just aiming for as safe and effective as? Which opened up a whole mindset change from our perspective. Let's change our mindset from replication to substitution. So when we think of that mindset change, it also is built upon a basic premise that a well-defined medical device that is easy to use and fits well with the user's workflow, whatever the context of use, can improve the clinical effect by reducing the likelihood of use error. So improving patient safety and potentially leading to better treatment outcomes. Shifting our mindset changes the approach from as good as to safe, effective, and substitutable. It encourages innovation and incentivizes improving user experience, with the key piece here being mitigating known use issues that may be related to the RLD. It also allows us to embrace a risk-based approach, focusing on a generic product's unique context of use. And if you've looked at the 2017 guidance at all, as we start looking at changing our mindset, we found out that it necessitated the need to enhance the entire threshold analysis concept as presented in that 2017 guidance. The new mindset opens up new validation options. And that means, or that brings to mind the bridging study that was introduced in the 2019 guidance. And then, of course, a traditional human factors study as discussed in the 2016 guidance, as well as IEC 62366. And our proposal is to use 30 users per user group, where we have 15 RLD experienced users and then 15 RLD naive users or cohorts. Safe and effective use as our mantra has always been in medical device, safe and effective when used by the intended user in the intended use environment. Well, now we're suggesting it change slightly, safe, effective, and substitutable when used by the intended user in the intended use environment. Now, as we look at this next slide, the alternative methodology that we're putting forward is truly based upon a new product methodology. So a lot of these phases will look very familiar to you. And we're going to look at each one a little bit more in just a moment. So when we look at phase one, the goal of phase one is to align stakeholders on early design documents. Specifically, we're going to be looking at the comparative use specification and the comparative risk management plan. Now, you will notice that throughout this presentation, we use the use of comparative in the title of many of these documents. And that's really on purpose as a reminder that when we do a comparative whatever, the comparative component is to remind us to include any additional details as it relates to the intended substitutability of the device that we're working on for the RLD. So the intended substitutability of a generic for an RLD. We also want to make sure that any intended design differences are identified as early as possible, because we've now had the opportunity to compare and take a look at any known use issues from the RLD. We also want to include the unique context of use as it relates to RLD naive users versus RLD experienced users, and the potential for the negative transfer from those RLD experienced users. And then, of course, we want to make sure we take a look at the training expectations. As we look at a comparative risk management plan, this is basically the document where stakeholders agree on the methods for identifying and assessing potential risks and harms. We'll be using a CURRA later on, or a comparative use risk. Oh, and of course, it jumped right out of my head. I'm so used to the acronym, but we'll look at that in just a moment as well. Again, the use of comparative is as simple as it was before. We're including the intention for substitutability. We're included intended design difference, unique context of use, and any training expectations. Now, the one thing to remember is that delivery device constituent parts are the only thing in scope of what we're talking about today. Any drug component items, whether it's in the risk analysis, whatever, they're all out of scope. Now, when we look at phase two, we're looking at the threshold analysis. There are three separate assessments that are part of the threshold analysis. One is a comparative task assessment. The other is a comparative physical device assessment. And then the final one being the comparative labeling assessment. The following assessments, as we're going to be talking about shortly, are enhancements to those that are presented in the 2017 FDA draft guidance. So just so we're all on the same page, the threshold analysis as defined in the 2017 FDA guidance is a systematic assessment of the user interface. The user interface encompasses all components of the combination product with which a user interacts. This includes the delivery device constituent part, any associated controls and displays, as well as all product labeling and packaging. All design differences are evaluated for their effect on the user interface and subsequent clinical effect and/or safety profile. Now, if you look at the slide for just a moment, the 2017 guidance talks about no difference, minor differences, and other differences. Well, I'm proposing that we look at it on a scale from 1 to 5. And that'll make more sense when we revisit this topic a little bit later. 1 is no difference, 5 is other difference, and this minor difference encompasses everything in between. There will be some minor differences that, for all intents and purposes, you'd say there was no difference, but there really is a little bit of a difference. And there may be some minor differences that, as you look at it and think about, well, that's pretty substantial, but it doesn't really yet classify as another difference. So that's why this 1 to 5 scale kind of will be helpful later on. Now, when we first jump in, the first task is basically to create the generic task analysis. We've all done task analysis before. And the idea is we create a generic task analysis for the generic device, and then we reverse engineer the RLD's task analysis from its IFU. The next part of this phase, or this comparative task assessment creation, is to integrate those two task analysis into one comparative task assessment. Now, we want to use the generic as the basis for the foundation of this task assessment. And just as a side note, we're going to be looking at the details of a task assessment shortly. So just bear with me through some of the notes here. And then we're going to note any use tasks or steps differences, any sequence in task steps differences, critical task differences. Each use step or use task is evaluated using the perception, cognition, and action model to further identify any design differences. And then at the end, we'll categorize each one of those differences as no design difference, minor design difference, or other design differences. The resulting comparative task assessment is later used as the foundation for the comparative physical device assessment, the comparative labeling assessment, and of course, the comparative risk assessment. Let's take a look at what a comparative task assessment may look like. What we propose is that the first section of, excuse me, we do it in a spreadsheet. There are multiple ways that you can put it together, but the first several columns are the use tasks. So identify the use ID, your use task, step ID, use step, and whether or not it's a critical task. Pretty much just as we've always done. The next step is then to go ahead and look at your PCA analysis of each one of the particular components as it relates to the either the generic or the RLD. And you can see how for the generic online one, we perceived the method of blue safety release grip and removal direction. On the generic, it was a flip up and away from the injector, whereas on the RLD, it was a pull straight up from the injector. So we classified those as different, those difference classifications as other, and the classification for that was perception and action differences may lead to delayed use. Now, I won't be going through all of the tables in as much detail moving forward, but that gives you an idea of how it moves forward. The 2017 draft guidance refers to this as refers to this next document as a physical comparison of the delivery constituent parts. from my perspective, the comparative physical device assessment is substantially more straightforward than any of the other two. And it's a systematic and comprehensive assessment of the delivery device constituent parts. Say that 5 times fast. The generic and the RLD delivery device constituent parts are what actually are evaluated. And it's pretty straightforward. You just have to look, you analyze, you may do some engineering tests. There are multiple ways that you pull all the different information together. And we're going to look at an example of that assessment right now. So again, we start with the use tasks. Everything always ties back to the use tasks. So that's the foundation that we build on. Then as we move forward, we have the physical characteristics, and you can see how they spread across, and which gets us to the final physical characteristic assessment and the physical characteristic evaluation. I'll let you see those on the screen. And just as a reminder, you will have access to these slides as they'll be sent to you after the presentation today. The comparative labeling assessment is a systematic and comprehensive assessment of all labeling characteristics and differences between the generic and the RLD. This is probably one of the most complicated of the 3 assessments because it's a analysis of visual elements, and it just is. There's a little more to it. It's a lot more subjective. So the idea is it's a little more of a handful. But what it does do is provide insights into the potential user actions that may impact the clinical effect and or safety profile of the generic based on their perception and or comprehension of the labeling elements. When we look at this assessment, you'll see once again the use tasks always start, and then we look into the labeling characteristics of the standard use characteristic is listed in that first column there on the left. And then as you move across, it evaluates and assesses the IFU between the generic and the RLD, any on-demand labeling or additional labeling, and the product packaging, both between the generic and the RLD. You can see that they've been identified in this table as G for generic, R for RLD, just because there was so much information to squeeze into such a tiny space. The comparative risk assessment is a clearly organized integration of threshold analysis data and known use issues, potential use errors, and foreseeable misuse. Its purpose is to present a clear picture of the risk assessment and provides a clear level of traceability. In the end, the FDA or the agency will look to the comparative risk assessment to evaluate overall risk profile of the generic in order to support your claim of having a similar or better safety profile, being therapeutically equivalent to a reference product, and then also produce the same clinical effect, all without healthcare provider intervention and without any additional training. This specifically is for the RLD experienced users. All design differences are actually evaluated in the comparative risk assessment for their effect on the user interface and on subsequent clinical effect and or safety profile. We remember the reason we use the word comparative. I'm not going to go through those again, but the idea is as we look at the comparative risk assessment that follows, one thing to remember is once you get to this phase, you really do not want to make any adjustments to the critical task designation. Any changes to the critical task designation should have been made prior to the beginning of phase 3. As we look to the next grouping of columns, we look at the potential use error. We look at the steps leading to the potential as hazardous situation and so forth. Now, again, we pull each piece of information from, in this case, the first one is the comparative task analysis. We have the second row there is the comparative physical device assessment. You would also put in potential use errors. For example, a user attempts to remove a safety release with one hand, and you would include any foreseeable misuse. Moving forward then, this particular component is a bit controversial. Let me put it that way. The systems engineers aiding in the development of this methodology advocated for the inclusion of probability so that they could maintain consistency within the design history file as a key component of the risk analysis. In addition, some manufacturers may require probability to maintain compliance with ISO 14971 conformance, and also in alignment with their ISO 13485 quality systems. Now, if this is the case, we suggest your probability index will always be 5. And the reason we say 5 is the agency's stance concerning difficulty of accurately assigning probability to use errors in any occurrence is undesirable. So we want to assume a worst case scenario when it comes to probability for this particular piece. Now, when you're done, before you send your comparative risk assessment to the agency, we do recommend you delete these 3 columns. All it will do is complicate your submission, and let's keep it simple and provide the information as required. The last component is where we actually look at the external critical design difference, i.e. no design difference, minimal, other, and whether or not the risk is acceptable. The method we use for mitigation, and then of course the validation method that will be used. Now, as we start looking at validation, hopefully I'm not speaking too fast here. As we look at phase 4, the task here is to validate the generic system as being safe, effective, and substitutable when used by the intended user in the intended use environment, knowing that a generic could be substituted with or without a user's knowledge, which could potentially lead to a high likelihood of negative transfer. So ultimately, the validation needs to be able to have supporting data to show that the clinical effect is the same or better, safety profile is similar or same or better, and that RLD experienced users can safely and effectively use the substituted device without healthcare provider intervention and without any additional training. Now, the guidance provides basically one option in 2019. I'm sorry, in 2017, the comparative use human factors study. However, we're going to look at 2 others and the way that we've proposed implementing them. The first one is in 2019, there's a guidance that introduced a bridging study. And the bridging study was in the 2019 guidance specified for investigational new drug applications, and biologic license application for device combination products. It is reasonable to expect that a bridging study will also be implemented to support ANDA because of the previous history we have with the bridging study with the agency. The purpose of a bridging study is to establish scientific relevance of information. So that information may have come from an earlier phase of the development program. It may have come from another development program, or it may have come to support the combination, but in the end, it's to support the combination product as being safe and effective. The intent is to compare. In a bridging study, we're going to compare critical product attributes, and we're going to leverage any additional testing and/or data that we have access to. In the end, we want to support that any design or user interface differences are acceptable, and a bridging study should always be tailored to the user interface difference that needs to be assessed. Now, when we look at the traditional simulated use HF study, this is pretty much what we're all used to. It was presented in the 2016 guidance. It's documented in IEC 62366. It's the concept of studied design, safe and effective use of all critical tasks, and root cause analysis are all parts of this particular, it's all specified. Excuse me. The idea is we want to validate all generic critical tasks and any other design differences as are required. Root cause for all use errors, close calls, and use difficulties, and note any negative transfer items that may have happened due to those design differences. Assess all intended users, patients, caregivers, etc., and other product differences in their context of use. Standard sample size, we would recommend remember, we're looking at two contexts of use here, right? We have the RLD naive. We would recommend 15 users because we're evaluating for safe and effective use. For RLD experienced users, once again, 15 users, and we're evaluating for safe, effective, and substitutable use. There are some times that RLD experienced user groups cannot completely be represented, for example, children's or adolescents, because it's unrealistic to expect that, you know, children and some adolescents are actually providing their own care. That would probably be done by a parent or guardian. There we go. So phase 5 includes reviewing all usability tasks. All usability risks identified throughout the product development process and ensuring they are updated in the CURA, specifically those that were identified during validation testing. So what we would look at is it's important to thoroughly analyze all validation use issues for root cause and the overall impact that they may have on the risk profile. Should design changes be required, additional validation testing may be required to test those components. And utilize early formatives, just as we always say, utilize early formatives to mitigate the risk of having to make design changes late in the product's life cycle. Since the CURA is a living document, a final update and review is conducted to ensure that it is current and accurate in its final state. We want to evaluate close calls, use difficulties, as they provide nuance in assessing final, safe, effective, and substitutable use. This document will become the foundation for the determination of safe and effective substitutable use for both RLD naive and experienced users. This would be the final document that would be submitted along with your application. The comparative residual risk assessment, depending upon your organization, how your organization works, this is basically a written risk assessment report that would come from your risk management team. And this would then be an integral part. Excerpts of this report would actually be used in the following report that we'll look at in just a moment. So phase 6 is, yes, you guessed it, it's Appendix A from the 2016 guidance. It's a summary of all data gathered, generated, assessed, and/or analyzed to support the generic as being safe, effective, and substitutable for the RLD. Reports on safety related HFE-UE considerations, risks, process, resolutions, conclusions are all included clearly and concisely. Everything is well documented, clearly clearly presented, and if it's put together cleanly, clearly, it can lead to a relatively quick approval process. The key is to make sure you have enough detail to support, and then you have enough validation data to support the claim that the generic is safe, effective, and substitutable. Straightforward, the level of detail should be sufficient to support the claim. If you're a little over wordy, that can sometimes just make reading the report a little bit tough. So think through the story, tell the story clearly and concisely. And then the following slides here that we're going to go through very, very briefly. You will receive, as I said before, you will receive copies of this presentation. And the idea is wherever we see the italics on the screen, as we see here, those are the changes between the Appendix A from the 2016 guidance and the proposal we're presenting here in a comparative HFE-UE report. Like I said, I'll go through these very, very quickly so we can get to the question and answer part. But the information is all pretty straightforward. It's pulling information from your comparative assessments that we've completed, the comparative risk assessment, and your validation data. Section 7 is on the long side, so I will skip over most of it, but it does bring in all of the enhancements as it relates to the bridging study, the more traditional human factors validation testing, and so forth. Now, the goal of the research, the goal of this process that we've tried to put together was to provide efficiencies and reduce the burden of the ANDA process. We wanted to simplify the process of bringing generic products to the market. The methodology that we're putting forward aligns with usability engineering best practice. It aligns with FDA 2016 guidance, and it aligns with IEC 62366. We've enabled innovation, allowing for resolution of known use issues and improved user experience. Hopefully, we've clarified the concept of unique context of use between the RLD naive and the RLD experienced users. Simplify the process by providing guidance on how to complete a threshold analysis and the enhanced risk assessment. Provide alternative methods for validation, and greatly simplified the requirement of a comparative use human factors study where I don't know if there's really any upper limit, but it's pretty much accepted that it's a minimum of 120 users per user group. So anywhere from 120 users plus, and you can figure out the difficulties or the time and labor commitment to running a study of that magnitude. In the end, the goal is to stabilize patient access to life-supporting medicines that are safe, effective, and substitutable. Nicole, do we have any questions?
We do actually. Great job, by the way. Thank you so much.
Thank you, Nicole.
Okay. I have a question here, may or may not be relevant, but can you talk about biosimilars at all? And do you think the FDA will ever allow biosimilars to be used in a different route of administration?
I don't really feel comfortable commenting on that in this type of an environment. But I would be willing, my email address is here. I would be willing to take that one offline if whoever asks it reaches out.
Great, no problem. And just a reminder, folks, if you have any questions for David, please feel free to put your questions in the Q&A function at the bottom of your screen. I do have a few questions, and here is one for you, David. How do you anticipate these methods simplifying the process of bringing generics to market?
Sure, that's a really good question. And the idea is, if you look at the process as it currently stands in the 2017 guidance, it's not very clear. So what ends up happening is when you try to implement without really clear guidelines, you end up running around a lot. That's one piece. So first of all, this alternative method is allowing each manufacturer to analyze the data and report results appropriately. The heaviest part of the burden, from my perspective, is in the 2017 guidance. We're basically only given the option of a comparative use human factor study. Now, as I said before, when you look at a comparative use human factor study, you know, there's even stories where the agency has come back and said, you know, you need more participants. So the point is, if you go with just what's accepted as pretty much the minimum, and I can't say it's the minimum, right? But it's pretty much generally accepted. The minimum is 120 participants per user group. So if you have 2 different user groups, you're talking about roughly 240 participants. Now, add into that over recruit, add into that potentially having to go to multiple markets, multiple regions, just because of the intensity of that recruit. It can take 2-3 months just to complete the fielding of that process. So the idea is for all intents and purposes, yes, the threshold analysis component is a bit of a challenge, but the idea is it's straightforward, it's well thought out, it's well designed. You end up with your differences. You know what's minor, you know what's other. You can bring that forward into validation. You have the option of using a bridging study. You have a much smaller 2-user groups. You're talking about 60 potential participants with an over recruit. That can be completed in 10 days for all intents and purposes, maybe 12. So it's this idea of really time. There's a big time savings, but we all agree that time is money. So there can be a great um, cost savings as well.
Awesome, thank you. I have another question here. I like the idea of a mindset shift. How do you anticipate the agency will accept?
That's a real good question. As we've stated, these methods were put together under a grant with the FDA, and they are reviewing. I would propose that the methodology, because it's built upon 21 CFR 860, others, but the main one that we presented in the slides here, 21 CFR 860, IEC 62366, as well as various FDA guidances that are published. The guidances are interpretation of relevant CFRs. So the idea is, I submit that the agency is really more interested in making sure they're getting safe and effective and substitutable products on the market than they are of keeping them off the market. So in this case, there are two approaches that you could put forth. One would be to present a usability engineering plan to the agency in advance, have them review your plan and approve it or not, get their comment as to why. The idea is just make sure the usability plan is complete and thorough and clear, right? And then the other thing is, my gut is that and my experience has always been with the agency, it's not, they may push back, but they don't push back for the wrong reasons. They always push back for the right reasons. So the idea is just make sure that you have a well thought out, clear, and I'm going to use the word story that you're telling. Make sure it's backed by data, not this idea of all these pieces. Hard data is what will breed success when dealing with the agency.
Yeah, really good point there. Thank you. Okay, I have one more question in the queue. So folks, just a reminder, if you have any questions for David, please feel free to put them in the Q&A. Okay. Innovation on a generic. Really? How's that possible?
I love that one. My goodness, I'd like to meet that person. Yes, the concept of innovating on a generic kind of seems unusual, because it is drilled into us that a generic is basically the same as the non-generic, right? And I think a lot of that comes from when we go to the pharmacy and we get a generic pill, they say its effectiveness is the same, right? And when it comes to a pill, it's a pill, we're going to pop it. the idea is when we're talking about innovation on a medical device, specifically a generic drug combination device, the problem is always going to be, can a RLD experienced user pick up that device and use it without thinking and go forward? Now, what we're proposing is a well-designed medical device that is easy to use and fits in the workflow, no matter the context of use, whether they're a new user or an experienced user. It can improve clinical effect. So the idea is, if this is kind of a hard one because it's easier if you have a visual picture in your mind, or you have a device that you can hold and you can think through, right? But for example, there's a product on the market that if you go into mod, there's a lot of people who use the device upside down and they end up injecting their thumb. Not good, right? So what would happen if you redesigned the device a little bit so that when you grabbed it, you knew which was up and down, right? Just from the affordance of the device. And then through product labeling, different components there, you're able to improve the safety of the device and make it just as easy for experienced users to use, and potentially easier for non-experienced. That's the piece. That's why this innovation component is so important in this particular methodology. Is it's the idea of driving the right change into products that may have been released so long ago that they're just kind of accepted for all their flaws.
Really good point. Thank you, David.
Thank you.
All right, folks. Well, that's all the questions we have for today. If you have any questions that you didn't get a chance to ask, you can see David's email on the screen, or you can scan the QR code. As David said, you'll also get a copy of this webinar and the slides after. So feel free to reach out to them if something comes up after the fact. With that in mind, David, I'd like to say thank you so much for your time and your expertise.
Thank you, Nicole.
Very interesting. Thank you to everyone who joined us live today. We appreciate your time as well. And thank you to Battelle for sponsoring today's content. We cannot put on these great educational sessions without support of our members and sponsors like you. I would encourage everyone to go to massmedic.com/events and view all of the fantastic events we have coming up. And we'll see you next time. Thank you so much. Have a good day.
Thank you, everyone.